Hereby I open this academic ceremony at Maastricht University. First of all, I welcome Mrs. Femke Ehlers, and she will defend her thesis in public, which is called Cancer Immunotherapies, Challenges and Opportunities for NK Cells in the Tumor Microenvironment. Welcome to all members of the degree committee. I will introduce now the two supervisors. Professor Gerard Bos, he is professor of internal medicine, in particular immunotherapy of cancer, and the other supervisor is Dr. Lotte Witte, and she is associate professor of transplantation immunology at Maastricht University Medical Center Plus. I will introduce the 6 opponents later during the ceremony, and one of the opponents is not here, but he is present online. Welcome here in the aula, and also welcome all followers of the live stream. I would like to invite you to give a summary of your thesis, but I know that there is a technical problem, and that means that the screen that you see here as a black screen doesn't work. There is a possibility to follow the presentation if you use the live stream, and I hope that you have got the link. It is somewhat, the presentation is then somewhat retarded, so it may take 1 or 2 seconds before the next slide appears, but you can follow the presentation on your smartphone. We are able to see it on a little screen here, and there's a screen as well, but then you have to turn around and we can see it quite easily. Mrs. Ehlers, may I ask you to give a summary of your thesis, and I wish you lots of success in the coming hour. The floor is yours.
Dear Prorector, dear committee members, dear members of the committee, dear colleagues, friends and family. Welcome to the defense of my PhD thesis. In the next 15 minutes, I want to give you a summary of the work I have done and how we can use NK cells for immunotherapy. As you know, cancer is a major health problem, and although treatment options have improved, unfortunately still a lot of people die from this disease. And as you can see in this graph, cancer is the second most common cause of death in the world, with numbers increasing over the years, and that have reached 10 million cancer deaths in 2019. Current common types of therapy include surgery, so removal of the cancer tissue from the body, as well as radiotherapy and or chemotherapy. But these therapies can come with severe side effects, as you might know from chemotherapy. And also, these therapy types are not always effective for all cancer patients, and especially if the cancer has spread to other parts of the body. And there is a newer type of therapy that we call immunotherapy, and this therapy type helps the immune system to fight cancer. So our immune system is a highly complex network with many different cell types present that protects us every day from infections like virus infection, bacteria, etc. And we can now use these functions for therapy to fight cancer, and this is what we call immunotherapy. There are many different ways of immunotherapy, but they all have in common that they activate the immune system so that it can better detect and destroy cancer cells. One way of immunotherapy is using drugs, such as signaling molecules or antibodies, and these are used to activate the patient's own immune cells. But there's also another way of immunotherapy called cell therapy that uses different types of immune cells, either from the patient itself that can be isolated, or from a healthy donor, which can then be isolated and given to the patient. And in our group, we focus on NK cell-based immunotherapy. NK cells stands for natural killers, and they are one type of immune cells present in our blood. And as their name suggests, they can kill cells. For example, virally infected cells or cancer cells. And to do that job, NK cells are loaded with killing molecules that I indicated in blue on my screen, but you can't see right now. And to prevent that NK cells kill healthy cells express a stop signal that then tells the NK cell to stop the killing process. Whereas NK cells can kill cancer cells because cancer cells express different molecules that then tell the NK cell to start the killing process so that the cancer cell can be killed. In this example, I only talk about one stop signal and one activation signal. In reality, it is a lot of different signals that the NK cell receives, and then it is the balance that decides whether an NK cell is stopped or activated to kill. But also, some cancer cells can escape this NK cell-mediated killing and tip the balance back to stop the killing. And they can, for example, do that by expressing the same stop molecules that normally healthy cells express. And because of this problem, that cancer cells can escape the NK cell-mediated killing, our group is focusing on different strategies, how we can reverse this to eventually develop successful NK cell therapies. And in our group, we focus on donor-derived NK cells. So we isolate the NK cells from the blood of healthy donors. Then we can culture these NK cells in our laboratory with the idea to eventually give these cells to the patient. And in our research group, we focus mostly on two cancer types, namely breast cancer and multiple myeloma, which is a form of blood cancer present in the bone marrow. And culturing the cells in our laboratory also gives us the opportunity to study different strategies, how we can actually make them better killers and optimize their killing capacity, and thereby shift this balance back towards NK cell activation and eventually tumor cell killing. In my thesis, I have tested two strategies to do so. First of all, to remove stop signals, and second of all, to add more activation signals as two strategies to shift this balance. Removing stop signals can, for example, be done by selecting NK cells from donors based on a genetic factor. And for that, you have to know that the main stop signals is a genetic factor that we call HLA. And within the patient, the NK cell receives this stop signal, and we call this a match because of a match of the signals. But we are all genetically different from one another. And for NK cell-based therapy, we can actually use these differences by selecting a donor with the different genetic factor HLA, and this NK cell from the donor will then not receive or recognize the stop signal coming from the cancer cell. And without this stop signal, it will be easier to shift the balance towards NK cell activation. We've previously shown that this strategy works for multiple myeloma, and I've now tested it for breast cancer. And what we then saw is that in the mismatch situation with an NK cell from a donor, we see higher NK cell activation than in the situation where there's a match situation of the stop signals. In addition, I added more activation signals by using antibodies. And antibodies are structures that can bind cancer cells, and by doing so also guide NK cell to the cancer cell. And this binding of an NK cell to the cancer cell through an antibody is known to induce very strong NK cell activation. And in my thesis, I have now combined these two strategies. So if I add an antibody to the match situation, then we see that there is a further increase in the NK cell activation if you look at the red bar compared to the yellow. If I combine these two strategies, the mismatch and the antibody, And the fourth bar, we see a further increase of the NK cell activation, suggesting that this combination can be a strategy to tip the balance towards activation. So far I have shown you that we can optimize the NK cell killing capacity by removing stop signals, by selecting donor NK cells with a genetic mismatch. And in my thesis, I have also demonstrated that this especially matters if the cancer cells express high levels of this HLA. We can add activation signals by adding tumor targeting antibodies. And I've shown you that the combination of both strategies is promising to maximize the shift of the balance. But we know that cancers are even more complicated because they reside in a so-called microenvironment. So tumor cells, cancer cells, they're not there alone, but they're embedded in what we call tumor microenvironment. And there are also many other different structures present, like vessels, for example, blood vessels that then infiltrate into this tumor, and also other factors and cells that can support the tumor to grow. And paradoxically, sometimes even certain cells of immune cells that can support the tumor growth. So there are many different factors known to characterize this tumor microenvironment and also to block immune cells from being able to kill the cancer cells. And some of these factors I have studied in my thesis. So, for example, it's known that there's often low glucose levels, so low sugar present. And that is as a consequence of the tumor cell growing, because every cell in our body needs sugar to grow and to function. As a consequence of the tumor cells growing, there's often low glucose. Another consequence of the tumors growing is often low oxygen, because the blood vessels cannot so well reach every cell anymore and distribute the oxygen. And it's known that these two factors can inhibit NK cells in the patient as well as other immune cells. And that is why I studied whether and how these factors also affect the donor NK cells that I want to use for therapy. So we have cultured NK cells and tumor cells together, either under normal glucose concentrations or under low glucose. And because glucose is so important, we actually expected that the NK cells would do less good under low glucose concentrations. But we did find that in both concentrations, in both situations, there was a similar level of cancer cells being killed by the NK cell. The same we've done for hypoxia, to culture the cells under normal oxygen concentration as a control or under low oxygen concentrations. And also then, we did not really see a difference between the two groups, which is actually very good news that the NK cell can still function under these environmental conditions. And it might suggest that the donor NK cells that we want to use for therapy might work better than the NK cells that are present within the patient itself. And then there's one other factor I would like to talk about, namely blocking the entry of immune cells into this cancer tissue. So for that, you have to know that cancer is a really dense tissue because there are a lot of cells growing in a very small area, which can sometimes make it difficult for immune cells to enter into this cancer. And this is why I studied in one chapter how this is for NK cells, how many can actually be there and where they are located. And for this chapter, I have used tumor tissue from breast cancer patients and then visualized the cells that are present within this cancer. And then you can see that every blue dot represents a cell. And in comparison to that, there are very few NK cells present that are here highlighted with the white arrows. But we also saw that in the same area, there are actually a lot of other immune cells present, here indicated in light blue or in yellow around the cancer. And when we quantified this, we saw that of all immune cells, it was only 2% of NK cells that were present, while there were many other immune cells present. So we found these few NK cells, and for the future it will be interesting to study why there were so few, whether they could not infiltrate into the tumor, and if it might also be related to these other tumor-supporting immune cells that were there, because we found so many other immune cells, and it's actually known that subsets of T cells and also myeloid cells can inhibit the NK cell function within the tumor microenvironment. And with that, I would like to come to the summary of my talk. So by now, we have multiple strategies available to create successful NK cells. And in my thesis, I've shown you two different ones, namely selecting donor NK cells with a genetic mismatch, and tumor-targeting antibodies to activate NK cells. And I also believe that the combination is key to successful NK cell-based immunotherapy. I've also shown you that the tumor microenvironment needs to be considered for NK cells to be successful. But I've also shown you that low glucose and hypoxia might not be a problem for NK cell-based therapy. But few NK cells in breast cancer were there while many other immune cells were present. So that's why I think that remaining challenges for the future will be to find the best combination therapy for each cancer, because we know that each cancer and each patient is different. And we should probably also enhance the NK cell infiltration into solid tumors. And with that, I would like to thank you for your attention and for your support during my PhD, and I give the word back to the prorector.
Thank you for your clear presentation. The opposition will now be opened by the chair of the Thesis Assessment Committee, Professor Axel Zurhauser. He is professor of pathology and he is affiliated with the Maastricht University Medical Center Plus. Professor Zurhauser.
Well, thank you very much. First of all, I would like to congratulate you and the team of promoters for this excellent book. Very exciting news. So, and to start with my opposition, actually, I want to give you a little pause and ask one of your paronyms to read out, I think it was Proposition 7.
Remaining challenges of immunotherapies include tumor resistance mechanism and the complexity and costs of manufacturing the immune cells for adoptive cell therapy.
Thank you very much. As propositions are supposed to be polarizing and challenging, may I ask you, what is the polarizing and challenging part of your proposition number 7?
Highly esteemed opponent, thank you for your kind words and for your question. So in this proposition, well, I think that the tumor resistance mechanism is still a challenge that we need to solve for NK cell therapy, and that will depend very much on the patient and the cancer itself. So it's the personalized therapy that we probably need to go for. And the complexity and costs of manufacturing these immune cells for therapy is still also an outstanding question that we need to solve.
Thank you very much. With this having said, I'd like to draw your attention to page number 78, chapter 4, and about what is ADCC, inducing antibody, trastuzumab, etc., etc. Long title. And on page 78, actually, you refer to HIF-1 alpha, and you say, in another study, hypoxia identified by HIF-1 alpha or HIF alpha, or measured by this expression. My question would be: is this enough to indicate hypoxia alone, or are there possibly alternative pathways leading to a HIF alpha or HIF-1 alpha expression not related to hypoxia?
I'm not sure because, yeah, I know that there are different markers to stain for hypoxia, yes, but so, yeah.
Alternatively, you can also answer the question if you would have preferred to have another marker added to this.
Yeah, exactly. So I think HIF1 alpha is not always sufficient to stain for or to visualize hypoxia. So there's, for example, also carbonic anhydrix 9, CAIX9, that I've used in chapter 5 to visualize hypoxia. Yeah.
Mm-hmm. And you talked about in your introduction about low glucose or let's say hypoxia. Are you basically talking about the Warburg effect? Is this situation the Warburg effect?
Yes. Yeah.
So why didn't you name it like this? What was the reason not to name it Warburg effect?
Not sure if there was the Warburg effect is the name for the cancer metabolism, right? So that the cancer cells in a low glucose environment change, or in the cancer environment change their metabolism to also still be able to grow and function in an anaerobic environment. Yeah.
So basically, is then the conclusion right to say that your assay only works if Warburg effect is present, or is this too far-fetched?
My assay only works.
Normoxic tumors would also respond, or
normoxic tumors would also respond, yes, and hypoxia is in it also. We have shown previously that hypoxia can also actually have an effect on the NK cells. So that was one thing, but then with cytokine stimulation, we can overcome this effect that NK cell can function in a low oxygen, in a hypoxic environment. And of course, it can also have effects on the cancer cell,
Very good. I'd like to turn the page to page 81, actually, and this is more a technical question, actually. You said that the primary human breast cancer tissue was obtained by us, by the pathology department. Mm-hmm. Was it how to formulate this best? Is it directly from the operation room to you?
No, so there, logistically, I have picked it up from the pathology department. And I do think that one variable factor there might be the time that it took from taking the tissue from the surgery to the pathology department. Of course, it would be more ideal if I can get it as soon as possible. But this was a bit of a logistic question.
I like this approach of avoiding the following question, but I have another one. It's about, is there a histology made by this biopsy before you assess or you used it for your assay?
Not yet, because this was a bit of proof of principle study that we just optimized the protocol to first of all get the single cell suspension, enough living cells that we can still do our assays. But I do think that it would be valuable to, in a follow-up study, to include, for example, histology, to have a better idea about the breast cancer subtype, etc.
I was more interested in the number of lymphocytes in there, but anyway, I'm very happy with the answers, and I return the words to the prorector.
Thank you very much, Professor Zuhause. The opposition will be continued by Dr. Eva Koenig. She is principal investigator tumor immunoediting, and she is affiliated with the St. Anna Children's Cancer Research Institute in Vienna, Austria. Dr. Koenig. Please switch on your microphone.
Is on.
Esteemed opponent, thank you for your question. Yes, so the discrepancy between degranulation and killing could be due to different mechanisms, I think. So that we see the degranulation, and especially in chapter 4, in combination with trastuzumab and the SKB3 cell line, we saw very, very potent degranulation. It suggests that it's not the NK cell not functioning, because it could kill the K562 cells, for example. So I think it must be a tumor resistance mechanism, and that can be several. So they could either be resistance to the granules, so either to perforin or to granzyme B. For example, Serpin is known as a granzyme B inhibitor. It could also be anti-apoptotic pathways within the breast cancer cell that they don't die. Because we do see kill, but not as much. I think another interesting approach could be the immunological synapse. And there was some papers that breast cancer cells also can remodel the actin shield and actually use it as a shield within this synapse, so that these granzymes cannot enter into the breast cancer cell. So I think there are many possibilities, and we need to investigate further, what is the reason? I think that we definitely need to include that more to study the level of the HLA-E, because indeed we showed in chapter 3 that the level is really important, and as far as I know, this is not always taken into account for the clinical trials. And I forgot the second half of your question.
If there is a difference between
well, actually, this week I've seen a paper that yes, it can be different. That there was an example of pancreatic cancer where the primary tumor had very low HLA-E levels, but as the cells were metastasizing, so these circulating tumor cells actually had higher levels of HLA-E than the primary tumor, suggesting that especially in this phase it's interesting to use monalizumab. And I think also there we should understand better how this HLA-E upregulation, what it could be caused by, what is the underlying mechanism. And I was thinking to myself, because HLA-E presents leader peptides of the classical HLA class 1, so maybe it could be related to that there's a difference in the HLA class 1 expression in the environment as soon as the cell leaves the primary tumor. But this should be further investigated, I think. Unfortunately, not yet. I would have liked to confirm basically what we see in the cell lines on the primary cancer cells, But we were not able to do that, partly because we often saw very low degranulation in the first place, so then it's difficult to see a decrease. And partly it was also related to the number of cells that I had available, because to determine this kill ligand mismatch, we do need quite a lot of cells per condition. So unfortunately, we don't have that data yet.
The material, would it work better in a metastatic setting?
Well, I hope so. And especially if we also then see in the metastasis a different HLA profile than in the primary tumor. So if we see higher HLA or at least HLA expression, then yes, I would expect that the kill ligand mismatching in combination with ADCC triggering will lead to a beneficial effect.
Thank you very much.
Do you have another question?
Would I have time for another? Yeah.
For a small question with a small answer, yes.
Question. Can you please very briefly explain bi or tri-specific antibodies or engagers? And I have a very specific question that I don't know the answer of. Do you think that this tri-specific antibodies or engagers, do they need to engage with all three epitopes in order to induce degranulation?
So bispecific and trispecific antibodies, they are not only targeting one antigen, which monoclonal antibodies does, but for bispecific 2 targets, or for trispecific 3 different targets. And there are actually many different people working on different targets to once engage the NK cell and on the other can't engage the tumor cell. And sometimes also in combination with the cytokine involved in this antibody to induce more potent proliferation and activation of the NK cells. And I'm also not sure. I would expect that we do need engagement of both for it to work, because that's also what we see with the monoclonal antibodies, that they target the cancer cell and the NK cell before it has an effect. So yeah, in my opinion, you would need both, but I'm also not sure.
Thank you very much.
Koenig. The next opponent is Professor Patrick Derksen. He is Professor of Experimental and Preclinical Oncology at the University Medical Center Utrecht. Professor Derksen.
Thank you, Prorector. Dear candidate, first I would like to congratulate you on this magnificent piece of work. I would also like to extend my congratulations to Dr. Wieten and Professor Bos. I thoroughly enjoyed reading your thesis, especially given the current collaboration with your former lab and my substantial lack of knowledge when it comes to NK cells, so that was good. So last month I got even scolded by an immunologist for calling an NK cell a T cell-like cell. So I don't want to get in trouble today, so I'll stick to what I know, which is breast cancer. So therefore, and perhaps not surprisingly, I would like to ask you a question about breast cancer in chapters 4 and 5. You mentioned breast cancer in your introduction on page 9, and even mentioned the fact that the disease is very heterogeneous, has several molecular subtypes, etc., etc. However, when I look at chapter 4 and 5, I just find a stratification between HER2 low and HER2 high. You seem to sort of maybe not disregard, but not look at the molecular subtypes. And in chapter 4, you're also stating, based on literature, that hypoxia can readily be detected in solid malignancies like breast cancer, and you're absolutely right about that. But it is also known that it very much depends on the breast cancer subtype. So if you have high desmoplasia, you usually have high hypoxia, and vice versa. So my question basically is, my first question, given the level of hypoxia, the heterogeneity in the breast cancer subtypes, what are your ideas on treatment towards breast cancer? Are there specific subtypes that would respond better to NK cell treatment than others, or not?
Highly esteemed opponent, thank you for your kind words and for your question. So regarding the looking at HER2 negative and HER2 positive, that was partly related to that we have trastuzumab available, the antibody targeting HER2 and known to induce ADCC. So we were particularly interested in that to see whether we can see a difference by combining NK cells with trastuzumab for the HER2 positive breast cancers and then compare it to the HER2 negative. But I do think that we can extend this principle to other antibodies to also target other subtypes, potentially anti-EGFR targeting. And yeah, you're certainly right that there are many differences between the subtypes and also in hypoxia level. So in chapter 5, I've actually seen also heterogeneous expression of hypoxia. So indicating that even if a tumor is hypoxic, it's of course not all regions. So it will be very local. And for that, it's important that NK cells can function in both environments. Regarding treatment, yeah, I do think that we need to activate the NK cells potently, but as we've also seen in chapter 4, for example, that's not necessarily enough. So I think we should combine it with other strategies as well to target the tumor resistance. And as I was elaborating before, we need to find out what the resistance is, but we could for example, target anti-apoptotic pathways to make the tumor cell more resistant to cell death. And I also think that it will differ for each subtype because, for example, HER2 positive and triple negative breast cancer are known to be more immunogenic than others. So if we go for NK cell therapy, either the adoptive cell therapy or stimulating the endogenous ones, it will be maybe easier or different to also engage a full immune response to.
Okay. Thank you, very nice answer. However, you're dodging the bullet a little bit. So I would like to go further on the subtypes, if I may, because you're using two types of cell lines as a breast cancer model. One is SKBR3 and the other one is MCF7. And they are completely different, not just from a HER2 point of view, but one is ductal, the other is probably lobular because of the E-cadherin inactivating mutation. Growth patterns are different. If you culture them in 3D, one is a solid ball, the other one is like a grape-like structure. So I would like to tempt you to do some predictions on what would be more feasible, which one would be more susceptible, because now you have an apple and an orange. One is low in HER2, the other is high in HER2. You see differences and then you conclude it's HER2. But it could be so much more. So how do you think that the subtype impinges on the effectivity of your treatment, given that one forms balls and the other doesn't?
So I think indeed one forming balls already suggests that the structure is different and that the tumor microenvironmental factors could be different. Mm-hmm. So that is certainly something that we then need to take into account to see whether hypoxic levels matter, for example, or also a nutrient, as well as NK cell infiltration, I think, because if it's a ball and there's less surface, there's less entry points for NK cells potentially than in a different structure. Mm-hmm. Yeah.
So do you envisage that if you have metastatic disease, that would be treated better than the primary tumor? Or is that a naive assumption on my end?
Yes, I think so, and even there are studies showing that NK cells might even be able to prevent the metastasis. For example, by the paper that I was referring to before, that HLA expression can be different, and if we can then target that at the right moment and give the NK cells, so yes, I think that could be one goal of NK cell therapy, to actually prevent the formation of metastasis.
Okay, thank you very much. I see some gentle hand gestures by the pro-rector, so I'll have a short question, if I may. Yep. It's about the kinetics of these NK cells. Again, I know nothing about NK cells, so correct me if I'm wrong. But what I see in literature is that most people do everything in suspension. They do a 1-in-1 or a 4-in-1 ratio, If I look at a solid tumor of 1 cubic centimeters, that's about 1 billion cells. If someone comes into the clinic with metastatic disease, that might be 20 billion cells. So you would need to add 80 billion cells to be effective. Is that even feasible? And what are the adverse side effects of something like that?
Well, I do think that by now we are able to generate a lot of NK cells, and many clinical protocols actually give 10 to the power of 8 NK cells per kilogram. So that is a lot of NK cells.
Okay.
But I also think that as we are learning more about NK cell biology and the expansion methods, that we can work on making one NK cell a better killer. So that one NK cell can hopefully kill more tumor targets that it can do at the moment, by, for example, stimulating the metabolism or making them more into a so-called serial killer.
Okay, thank you for your wonderful answers, and I would like to give the floor back to the prorector.
Thank you, Professor Derksen. The opposition will be continued by Professor Dirk de Ruijser. He is professor in radiation oncology and respiratory oncology, and he is affiliated with the Maastricht University Medical Center Plus. Professor de Ruijser.
Thank you very much. First of all, I would also like to congratulate you and your whole team and also your family for this wonderful thesis, because I know that the family is also, let's say, suffering sometimes for PhD thesis. So congratulations for that, but I'm here to ask you some questions. And actually, when I read your whole thesis, and especially chapter 3, you say that basically NK cells may work if you just give them the right trigger. That's basically what you say throughout your thesis, whether it's hypoxia or no hypoxia, doesn't matter. Now, I'm a clinician and I want to ask you, how would you start a clinical trial in a solid cancer patient right now? Because most of the time what we have seen until now is just a kind of agnostic approach. You just give something and we see what happens, basically. Put it a little bit black or white. But how would you now start? Not considering any financial limitations, of course.
Highly esteemed opponent, thank you for your kind words and for your question. If I were to start a clinical trial, I would definitely like to start with better characterization of the cancer and or the tumor microenvironment, because, like you said, that will give us a better idea what we need to target, so that we don't try a combination and then see whether it works or not, but that we, for example, assess hypoxic levels, HLA levels that we showed are very important for the NK cell function. And we can do that, I think, either by starting, for example, with a genetic screen, so take some DNA, because that gives us a lot of information, but I do think that we also always need the protein levels. And I think for that, the methodology that I used in chapter 5, so the multiplex immunohistochemistry, can be a very powerful technique, although you do need a biopsy of the cancer patient. But there we can then visualize factors and immune cells, where they're present and where they're located, to better know what we can target.
And would you then consider to take one single biopsy, like in lung cancer, for instance? Most of the time we do an endoscopic procedure and we take a quite tiny part of the tumor or a lymph node or something. Would you envisage that would be enough, or would you like us to take more biopsies?
Yeah, ideally from the research perspective, I would like to have more biopsies, but there we of course also have to consider the impact on the patient. But for sure, a small biopsy will not be able to tell us as much. And in chapter 5, I was lucky to have the quite big pieces of the primary tumor, and then we see even on one tissue slide a lot of heterogeneity in, for example, HLA class 1 expression. So suggesting that one biopsy will not be enough because it only gives us one small piece of information, and also at a certain time point.
And what about liquid biopsies or even imaging markers as a kind of biomarker? Have you thought about that?
Yeah, that could for sure be an option that we can include. So liquid biopsies, we would have to see how representative it is, but for sure we could check for factors that are present or potentially even already circulating tumor cells. Yeah.
And you always mention then hypoxia, also on a histological level, but is it really important? Because if I read chapter 3 carefully, I hope I did, then actually it doesn't matter. Because if you give the right antibody, you can activate whatever the subtype of NK cells are, you can activate them. So is it really important to look at hypoxia?
Well, we show indeed that the NK cells are not bothered by hypoxia, but of course it is an in vitro study, and we also only included hypoxia. So I do believe that it can get important if we have a combination of different factors. And the same goes for the chapter 6, where I was looking at glucose levels. I think the combination of different factors can still have a bigger effect on the NK cell, for example, the metabolism and also directly the function, than single factors. So that is why I think that we should still always consider hypoxia and actually confirm that also in vivo they can still function.
If I have time for last question, I think it's logical that you would try to combine an NK-directed treatment with another, let's say, targeted treatment. But did you consider, the old chemotherapy? Because we have seen in classical now, anti-PD-L1, for instance, it works pretty well with classical platinum-based chemotherapy. Would you consider this as well, together with NK-directed treatment? Also, how would you then envisage to combine this from a mechanistic point of view?
I think it could be interesting, but for chemotherapy, I think it can go both ways depending on the type of chemotherapy and on the dosing, that it can have inhibiting effects on the NK cells. So that's of course not what we want, but if there are low doses given, it can lead to NK cell activation as a response to the stress and that the chemotherapy triggers. So I think in some situations it could be useful, and then we would have to see that we give it in a way that it doesn't inhibit the NK cells. So presumably giving the chemotherapy first so that we can get the effect of the chemotherapy, the release of the activating ligands for the NK cells before giving the NK cell therapy.
Thank you very much. I give the word to the pro-rector.
Thank you very much, Professor de Ruijter. The next opponent is Dr. Sander Rense. He is Associate Professor of Surgery at Maastricht University. Dr. Rense.
Yes, thank you. Dear candidate, first of all, of course, I would also like to congratulate you on this really impressive piece of work and extend the congratulations to the team, because it's all about team science nowadays, and it is a big team and it was a good team. I really enjoyed reading the work. I also learned a lot, because it seems like you didn't invite an NK cell expert actually on the committee. But I really learned a lot truly. And the NK cell part is not my first focus, but as you know, probably I'm working more on metabolism, and that is why my attention was drawn to chapter 6. So I would like to take you there, and especially start on page 148. So it's the first figure. And there you investigated the glucose levels in the bone marrow of patients with multiple myeloma. And what struck my eye, and actually what you see usually in patient studies, is that there's really a big variation. And I was wondering, first of all, if you can maybe try to find an explanation for this big variation.
Esteemed opponent, thank you for your kind words and for your question. Indeed, in chapter 4, we started off with actually measuring glucose levels in the bone marrow of multiple myeloma patients, because the concentrations were not known before. And indeed, we see a big variation, as we often do between patients. I think it might be partly related to the time point that the biopsy was taken, and whether or not, I'm not sure how it affects the bone marrow, but whether or not the patient was fasting or not, I was thinking about. So I think the time point could be one reason, and then of course also differences between patients.
Yeah, I'm glad that you bring up the point of the timing of the sampling, because I was thinking, but I'm not an expert in that as well, that, you know, people might be fasted because it's probably under surgery or anesthesia at least. So I agree that could be an explanation, differences in fasted state and fat state maybe. Could it also be that BMI or maybe cachexia would be involved in this?
I think so, yeah. So that could be other factors that, depending on the BMI, the levels are different, or the staging of the cancer. Yeah,
so there can be many reasons. At least it's
Exactly.
important that you try to model this, and that's what you did in the next studies. So if you look at figure 2, you tested different concentrations of glucose in your cell systems. And the lowest concentration that you chose was 500 milligrams per liter. And I was wondering why you chose that concentration as the lowest. Because if you look at the variation in the population, there are people who are getting to a lower level even. And you also mentioned that solid tumors can go as low as 20.
Yeah.
So why did you start with the 500?
So we started with the 500 because that is the lowest concentration that we observed in the patients that we measured for this study. So that was our main reason, because we wanted to mimic the concentration that is present in the bone marrow. And I do think that, I would have liked to include also a lower concentration. Especially after we saw that 500 milligram does not matter to the NK cell. Another reason why we didn't go lower in the first place is that we already expected an effect with 500. It's because glucose is so important for NK cells. But then we did see that in IL-2 activated NK cells or expanded NK cells, it actually did not seem to matter.
Yeah, I can imagine, and in hindsight, it's always easier, right? So that's clear. Then, looking at the figure, I always like that people depict the individual data points, so you can see a lot of things in such a graph. And you know, on average, it seems that it doesn't matter, the glucose concentration. I agree, except for the really high one. But you can still see also there's big variation. I was wondering if the same donors were always responding in the same way at each glucose concentration, because that's what I cannot see in the figure, but I'm really curious about that.
If I remember correctly, then it was mostly related to donor differences more than it was related to the glucose levels. And this is also something that we frequently see, that, it depends on the receptors present on the NK cell that are then corresponding to the ligands of the tumor cells. So for example, there's a big variation in NKG2A expression, for example. And also in this study, we didn't type for kill ligand mismatch, etc., or the activating ligands. So yeah, as I said, I think from my memory, it was mostly the donor differences and not so much the glucose differences.
Okay, that's interesting. So this variation is what you see more often.
Yeah,
for
sure.
And so going to panel B, what you see is that some people kill 5%, some kill 30%, some donor cells. Does that really matter? Because the cytotoxicity assay takes 4 hours, and I was wondering if you translate that to like the in vivo situation, where of course there's more time for killing. Do you consider that these differences are really relevant?
I think that they are for sure relevant. I agree with you that we only look at 4 hours here, but NK cells are known to be really quick in killing because they are already loaded with the killing molecules, so the granzyme and perforin, and ready to kill when they meet their targets. So they can actually kill within minutes to a few hours. So for the granule-mediated cell death, I think we for sure will see that in the 4 hours. Mm-hmm. I do think that there's also the killing through the death receptor pathway, so the FOS and FOS ligand, that might take more time. So if we would have done the assay for a longer time, we might have seen higher killing levels. But also it was mostly about picking up differences rather than looking at a longer time.
I understand. Time's up. Okay. Thanks for the answers. I give the word back to the prorector.
Thank you, Dr. Rense. The opposition will be continued by Professor Pilar Martinez, and she is Professor of Neuroinflammation of Neuropsychiatric Disorders at Maastricht University. Professor Martinez, and she is also Secretary of this committee.
Thank you, Prorector. So I would like first to compliment the candidate for the very nice PhD thesis, a lot of work, a lot of papers, most of them published, so it's remarkable. Also to of course, the promotion team. It has been probably a lot of work, also the supervision. So, and my question goes to chapter 7. So I think you have a little bit of every chapter of the book. And I am very interested in this chapter where you use also combination therapy. So you use immunotherapy with an antibody. You are targeting, in this case, multiple myeloma, that is very difficult to treat plasma cell cancer. And then plasma cells are cells that are very difficult to target because of the very low receptors or types of receptors on the surface. So it's difficult to find antibodies to target those cells. And now, in this study, you use antibodies against PD-L1. So I would like to know, why did you choose that approach, so to combine your NK cell with an anti-PD-L1 antibody? Is there a specific on those antibodies that make it more attractive to use for this type of a study?
Highly esteemed opponent, thank you for your kind words and your question. And in chapter 7, we wanted to focus on the other functions of NK cells than only the killing mechanism, which is the cytokine secretion, such as interferon gamma, which can also be very important to kickstart an immune response. Then we have chosen PD-L1 because it is already working, like it's already used in the clinic, the checkpoint inhibitors, but then we have chosen the variant that can also induce ADCC to basically see, because this is a checkpoint molecule, so it's not restricted to the cancer cells, but it can also be expressed on the tumor microenvironment, and as we, for example, see on the macrophages that we cultured. So that was basically the idea, to combine it with another target that is not cancer specific, but I do think that we can also, that it will not only work with anti-PD-L1, but could also work with other antibodies.
So, for example, anti-PD-L1, if you inhibit this, you could also get, for example, another type of disease as a consequence of your treatment.
Yes.
So what will that be? So if you get the opposite effect, so by your treatment, so which type of side effect could you get if you block these checkpoint inhibitors?
Well, I think checkpoint inhibitors are there for a reason, of course, to stop the immune reaction and to restore the homeostasis. So I do think that if we push the balance too much and create too much immune cell activation, that we might actually get the opposite and potentially autoimmune diseases, or at least reactions against the normal healthy tissue that we do not want.
Yeah.
Because, yeah.
So, and then I was wondering about that other antibody that is also used for targeting plasma cells, that is that Dumab, that is anti-CD38, sorry. And then what will happen if you use this antibody that target plasma cells to your NK cells? So do you think it's a specific effect of this antibody into the normal NK cell population?
I do think it does, because daratumumab indeed targets anti-CD38, which can also be expressed on healthy NK cell populations. So in that sense, we could have the side effects that NK cells are targeting each other through this antibody, and that is something that has also already been studied and shown. So this is called NK cell fratricide, NK cells killing each other. But we also have possibilities to prevent that. For example, now that engineering of cells is more popular and possible, for example, we can create anti-CD38 like CD38 knockout NK cells to knock out the gene so that NK cells are not affected anymore, or not as a negative side effect, but that it's really targeted to the target cells also.
And since I see the time, I would like to ask you, to your paranyms, to read the last proposition in your thesis. The last one, number 9.
There is no real ending. It's just a place where you stop the story, by Frank Herbert.
So it seems that you really didn't want to finish, and you had many things to do.
Yeah.
So I would like to ask you, so what did you miss to do in your thesis? So what would you have loved to do and you didn't have to time for? Sorry.
Well, I think a lot of chapters I would have loved to continue. So, for example, chapter 4, where we used NK cells in combination with the mismatch and the antibody for breast cancer cell lines, we saw the discrepancy between degranulation and killing. So there I would have liked to understand the mechanism, how the breast cancer cells escape the NK cell-mediated killing, and then to use that knowledge to combine it with another therapy approach. The same goes for chapter 5, where I used the multiplex immunohistochemistry technique to, first of all, stain NK cells and see where they are located. And unfortunately, I managed to use 20 markers, but even 20 markers were not enough for me, because I would like to follow up to see if the cells that we find are actually truly NK cells and what their functions are, because NKp46 positive NK cells could potentially also be the innate lymphoid cells, which have a slightly different function. And as well as understanding the tumor microenvironment better, because now it was rather a broad characterization than really understanding the mechanisms behind it. Yeah.
Thank you for your answer.
As you have seen and heard, the time for defending your thesis has passed. The committee will now withdraw to discuss the quality of your thesis, and in particular the quality of your defense. And please await our return here.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Long road to the south side, 7 miles in my rearview mirror. I know what it felt like. My goals only getting clearer. East side to the west side, no place like home. If it's questions that you've got, go the extra mile and that. On the line, I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile. Long road to the south side, 7 miles in my rearview mirror. I know what it felt like. My goals only getting clearer. East side to the west side, no place like home. If it's questions that you've got, go the extra mile and that. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Take the mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. And even though these nights seem long, I know that I am never on my own. Know you've won the battle once you've grown. South side, 7 miles in my rearview mirror. I know what it felt like. My goals only getting clearer. East side to the west side, no place like home. If it's questions that you've got, go the extra mile and that. Long road to the south side, 10 miles in my rearview mirror. I know what it felt like. My goals only getting clearer. East side to the west side, no place like home. If it's questions that you've got, go the extra mile and that. What's on the line? I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile.
in Zweden. En ik had dat eigenlijk graag willen doen. Daar schrijf je dan hier iets persoonlijks voor in. Denise zit alleen.
Ja, hier eventjes. Je wilde even front row zitten. Je wilt niet front row. Front row.
Mrs. Ehlers, the committee here present, and not only here but also online, has discussed the quality of your thesis and in particular the quality of your defense today. And in view of its positive verdict, it has decided to grant you the degree of doctor. And also we have taken into account your previous qualifications. And therefore, we will grant you the degree of doctor, and your supervisor, Dr. Lotte Wieten, is authorized to confer on you this academic distinction in accordance with Dutch University custom and law. I invite now your supervisor to take the floor.
Please all rise. Thanks. Okay, do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
Yes, I promise.
By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Femke Augustine Irene Ehlers, the degree of doctor, and grant you all rights attached by custom and law. As evidence of this, I now present you with the degree certificate signed by the rector, the secretary, and the other members of the committee, and affixed with the official sign of the university. Okay, and it's my great pleasure to also do the laudatio. And with an international person like you, it's always difficult to choose languages. We were just discussing, and then we said, okay, we do it in Dutch. So I'll switch. Zeer geleerde Dr. Ehlers, beste Femke. Het is mij een groot plezier om jou, ook namens Gerard, mede-promotor, te feliciteren met het behalen van jouw dokterstitel. Jouw werk heeft geresulteerd in een prachtig boekje. Op 1 hoofdstuk na is alles gepubliceerd. En dat is echt een hele prestatie met zoveel hoofdstukken. En in de afgelopen jaren heb ik jou zien groeien. Als wetenschapper, maar vooral ook als mens. En vandaag heb ik ervan genoten om jou je thesis te zien verdedigen. Want ook dat heb je prachtig gedaan. Heel overtuigend en super mooi gedaan. Jouw tijd bij ons op het lab begon in 2016. Toen je solliciteerde op een project over NK-cellen en kanker. Je gaf daarbij een presentatie over jouw stageonderzoek in het Karolinska. En dat was zoals we jou later ook hebben leren kennen. Het was heel solide. Je kon prima uitleggen wat je gedaan had en ook waarom. Het was ook down to earth, zonder franje en zonder grootspraak. En dat is heel kenmerkend voor hoe ik je de afgelopen jaren heb leren kennen. En ik denk ook voor hoe je vandaag hebt verdedigd. Je bent een echte teamplayer. Je kan heel goed organiseren. En ook dit is een constante door jouw hele PhD-traject. In het begin werkte je heel veel samen met Niken. Zij leerde jou de kneepjes van het NK-celvak. En jij hielp haar met het afronden, ja, ze is er ook, met het afronden van haar stukken. En jullie waren een heel complementair team. En het heeft ook geleid tot een heel aantal mooie studies die gepubliceerd zijn. En het is nog steeds de basis voor een heleboel vervolgonderzoek wat we aan het doen zijn. En ook voor voorbereidingen voor een klinische studie. Later ging het vaak zo dat Gerard of ik met iemand hadden gesproken en een half idee hadden en jou daarvoor probeerden te enthousiasmeren. Of misschien was het meer eerlijk het bij jou dropte. En jij zorgde er dan voor dat er ook een realistisch plan kwam. Dat de METC-goedkeuring er was, dat we uiteindelijk de data kregen, dat alles geregeld was, studenten gesuperviseerd waren. En je hebt heel veel labwerk zelf gedaan. Dat vond je ook leuk om te doen. Een paar voorbeelden. Je organiseerde samen met Loes van de pathologie de samples van borstkankerpatiënten voor hoofdstuk 4. En je zette een humaan in vitro model op. Samen met Timo, jouw paranimf, werkte je aan HLA-C-splicing. Een echt out of your comfort zone project voor jou. Je was mede-organisator van de faxcursus en de immunology seminars. Je was onze echte faxkoningin. Iedereen werd door jou geholpen. En met Christian heb je een uitstapje gemaakt naar NK-cellen in adipose tissue. Al denk ik bij die laatste wel steeds van, goh, ging dat nou echt om die NK-cellen? Of waren daar misschien ook andere factoren die daar een rol bij speelden? Want tegen de tijd dat jij ons vertelde dat je wat met Mitchell had, hadden wij dat eigenlijk allemaal lang in de gaten. Je was ook altijd in voor gezelligheid. Op het lab, tijdens pauzes, tijdens de borrels, tijdens de carnaval, niet te vergeten op congres. Je nam daarbij initiatief, je organiseerde dingen. Je was er altijd bij, in de rol als Chief Google Officer, CGO. Als Indiaan of met typisch Duitse, qua hoeveelheden, glazen bier. En ik denk dat bij de meeste congresparties of afterborrels jij ook degene was die het licht uitdeed. Natuurlijk altijd met hulp van enkele niet nader te noemen overige leden van ons team. Het is ook niet verrassend dat in jouw tijd in Marseille veel van de mensen met wie je samen hebt gewerkt bij jou zijn langsgekomen voor een wijntje. Halverwege jouw PhD-traject kreeg je een fellowship van GROW om een half jaar naar Lisa Cossens te gaan in Portland. En naast het een hele leuke tijd heb je ook een mooie dataset verzameld waar we vandaag ook wat over gehoord hebben. Waar we nog allerlei analyses op konden doen. Door COVID moest je eerder terug, maar middels allerlei online meetings heb je er een hoofdstuk van gemaakt. Je hebt zelfs je promotor overtuigd dat het echt in je boekje moest. Een prestatie op zich. En Lisa had jou heel graag teruggezien als postdoc in het lab. En ik heb je ook gestimuleerd om dat te doen. Maar ik was eigenlijk ook wel heel blij toen je uiteindelijk koos om bij ons in Maastricht te blijven. Want je hebt daardoor, naast de studenten die je eerder al had gesuperviseerd, Nikki en Amber een hele mooie start van hun eigen PhD-track kunnen geven. Je bent een doener en vond het fijn om bezig te zijn in het lab. En misschien waren daarom de laatste loodjes ook wel zwaar. Veel schrijven, maar ook verhuizen van Marseille, een nieuwe baan, een baan afronden, verhuizen naar Zwitserland. Maar het is nu echt klaar. Je mag heel trots zijn op het resultaat. Ik ben het in elk geval. En ik heb met heel veel plezier met je samengewerkt bij ons in de groep. En ik wil jou en Mitchell en je familie en vrienden ook namens Gerard graag feliciteren met dit mooie resultaat. En ik wil jullie ook Mitchell heel veel succes wensen in jullie verdere carrière en in Zwitserland of in welk land het ook mag zijn.
Dankjewel.
Esteemed Dr. Ehlers, beste Femke. Het is me een groot genoegen. Het is een very nice pleasure to congratulate you with your doctorate, and I do that also on behalf of the Board of Deans of Maastricht University. And maybe I may share some impressions of the committee with you. We have seen a very clear presentation, and we hope and we think that it also for a lot of lay people here in the aula it's comprehensible. And we also saw an excellent thesis, and the thesis represents also a lot of work. That was very clear. And we were also impressed by the quality of it. And also, the majority of the thesis chapters are already published or underway to be published. And your defense was very good. We saw that you could easily answer most of the questions, and you have also demonstrated that you have answered these questions and also took part in discussion with a very broad scientific perspective on the subject matter of your studies. And so we are very satisfied with your defense. And I would like to include in my congratulations also your two supervisors, Dr. Lotte Witte and Professor Gerhard Bos, sitting here on the other side of the room. You may be very proud of the thesis and all the work that has been done under your supervision. And of course, I would like to include in my congratulations your family. And I think your family is on the first row here. Your partner, your parents, your grandparents, I think, and other members of the family, congratulations with the result that we have seen today in the form of this thesis and this very good defense. And of course, I would like to include also in my congratulations your other family members, your friends, and I think also a lot of former colleagues and still present colleagues here in the aula and probably also in the live stream. I think now it is a moment, but you were, I couldn't ask you before, but I would like to ask Dr. Koenig, who is not present here live, but online, would you like to congratulate the young doctor?
This wonderful PhD thesis and defense. It's a very comprehensive and beautiful oeuvre, and you presented yourself as a versatile and highly productive researcher. Putting those nearly 7 papers into one coherent thesis in such a clear and thorough way is even more remarkable. I can only congratulate you and your supervisors for such a fruitful time and collaboration, and wish you all the best for your future.
Thank you.
Thank you very much. I would like to thank also all the members of the two committees, the Thesis Assessment Committee and of this Degree Committee, for your critical reading and for having interesting and scientific questions and having a good discussion. So thank you very much, and Maastricht University appreciates your input. Before I end this academic session, I would like to say that we are going to take a picture with you and the committee, and if the ceremony is ended, I would like to ask the other people here in the aula to leave the aula and to go to the reception room, which is the rafter, and I think that the beadle will show you where to go. It's not far away, but we will arrive a little bit later at the reception. And hereby I close this academic ceremony.
Look at it. Volgens mij had ik er al meer moeten zetten.
Nee,
kon je het beter zien als ik.
Nou heeft iedereen gezien inderdaad.
nu, hè? Niet
Ja,
te vallen.
ja, ja.